Dear students, let's move on to the topic Mendelian Genetics. Mendelian Genetics originated after the works done by Gregor Mendel, who is known as the father of modern genetics. He was born in Austria in 1822. In 1840, he graduated from the secondary school in his hometown with honors. Following his graduation, Mendel enrolled in a two-year program at the Philosophical Institute of University of Almans, where he studied physics and maths. He graduated from the program in 1843. In the same year, he began to study to be a monk or a priest at the St. Thomas Monastery in Brno. In 1851, he was sent to the University of Vienna at the monastery's expense to continue his studies in sciences. In 1953, after completing his studies, he returned to his monastery and started teaching at a secondary school. There, he stayed for more than a decade, and it was during this time that he began his experiments on inheritance, for which he is best known. Mendel's experiments. Around 1854, Mendel began to research on transmission of hereditary traits in plant hybrids. He chose pea plants, that is, garden pea plants, for his experiments. His research continued for many years, for as many as 8 years, and he used several thousands of pea plants for his experiments. In 1865, the results of his experiments was presented by him in front of a eminent group of people at the Natural Science Society, Brno. The Natural Science Society, Brno, published the results of his findings in their journal under the title Experiments on Plant Hybrids. After his presentation did not get wide acceptance among the crowd present there. So hence he was very disappointed and he did little promotion for his work. After his presentation in 1868, Mendel was elected as a principal of his school where he was working, and due to his administrative duties and his duties at his monastery and his failing eyesight, he discontinued from his experiments on garden pea plants, and he died in 1884 at the age of 61. So, rediscovery of Mendel's work. It was almost after a decade and more after Mendel died, that is in 1900, three biologists or scientists, or we can say botanists, Hugo de Vries, Karl Correns, and Erich von Teschermark, each of them independently duplicated Mendel's experiments. That means each of them, without knowing each other, they worked on the experiments of inheritance, and while they were searching for any back references, they found out the fact that all of their data and their general theory were published in 1866 by Mendel. These three biologists gave complete credit of the work on inheritance to Gregor Mendel, and the significance of his work, that is Mendel's work, was appreciated by the scientific community. Thus, Mendel's studies began to be referred to as Mendel's laws, and he began to be considered as the father of modern genetics. Mendel's experimental material. One of the main successes of Mendel's experiments was the choice of the experimental material. The experimental material that Mendel chose is Pisum sativum, or commonly known as garden pea plant, belonging to the family Leguminosae. This plant was a widely cultivated herb, becoming shrubby. It was an erect and climbing plant which grew up to 3 feet high. The plant showed all characters of the family Leguminosae. Go through the botanical description of the plant from this slide. Reasons for Mendel's success. Although several of his contemporaries did research on inheritance pattern in several other plants and even in animals, they did not find success as Mendel did. This was because of several reasons, some of which are mentioned in this slide. One of the main reasons was the choice of his experimental material, that is, the garden pea plant, which was an annual, naturally self-pollinating plant. This plant had several pairs of contrasting characters, that is, opposite characters. Mendel concentrated on only one character at a time. He concentrated on only one character at a time, that is, he studied only one character at a time, which was not done by his contemporaries. Most of his contemporaries studied all the characters of the whole organisms, which made it difficult for them to arrive at a conclusion. Moreover, Mendel kept an accurate record of all his findings. His knowledge in mathematics and his knowledge in statistical methods helped him for analyzing the results he got. Moreover, the characters selected by Mendel was present on different chromosomes, and all the 7 pairs of contrasting characters selected by Mendel showed complete dominance. The last two of the reasons of his success was not purposeful. Unknowingly, the characters selected by Mendel for his study were present on different chromosomes, and the characters showed complete dominance. Hence, he could develop the first two of his laws. As I have told you in the earlier slide, advantages of the experimental material was one of the main reasons for Mendel's success. Let us see what are the advantages of the garden pea plant which led to Mendel's success. The garden pea plants are self-pollinating plants which resulted in the production of true breeding plants. That is, the plants produced will be exact replicas of their parents. All offsprings will have the same character generation after generation. The second reason or the second quality is the pea plants exhibited a variety of contrasting characters. Contrasting characters means opposite characters which was very conspicuous or which could be very clearly observed. The third reason or the third quality is the shape of the pea plant flower, which facilitated self-pollination or protected the plant from cross-pollination. The morphology of the flower made easy artificial hybridization. That is, the flower could be opened very easily for emasculation. You know what is emasculation? That is removal of unripe stamens or unmature stamens. And also it was easy for dusting the desired pollen grain on the stigma of emasculated flowers. So the morphology of the flower was easy for artificial hybridization, and that also facilitated Mendel's experiments. The pea plants was growing very quickly, or its growth rate was very quickly. It grew very quickly, it flowered very quickly, and further generations could be raised within short time span. Hence, the experimental results could be collected very easily within short time span. And the cultivation of this plant did not require much space. That was also one of the qualities of that experimental material. Now let us see the 7 pairs of contrasting characters of the garden pea plant Pisum sativum. I am going to read out the 7 traits or the 7 pairs of contrasting characters that Mendel studied. First one was form of ripe seed, that is smooth and wrinkled. Second one, color of seed albumen, yellow or green. Third one, color of flower, purple or white. Form of ripe pods, inflated or constricted. Color of unripe pods, green or yellow. Position of flowers, axial or terminal. And the last character was length of the stem, tall or dwarf. You can see several alphabets which is given in capital letters in brackets. For example, color of flower P is given in capital letters and in brackets, which denotes that the purple flower color is the dominant character in the plant. We'll learn about dominant character in later classes. So in each of the character, the for ripe seed, smooth is the dominant character. Then for seed albumen, yellow color is the dominant character. For shape of the pods, inflated is the dominant character. For color of pods, green is the dominant character. So position of flower, axial is the dominant character, and length of stem, tall is the dominant character. So we learn about dominance, recessiveness, etc. in further classes. So here a diagrammatic representation of the 7 contrasting characters of the Pisum sativum plant that Mendel chose is given. You can clearly see the seed shape, round seed shape, wrinkled seed shape, seed color, yellow and green, pod shape, inflated and constricted, pod color, green and yellow, flower color purple, white, flower position, axial, that is on the axis of the leaves, and terminal, and plant height, tall and dwarf.